Today's my talk is the molecular mechanism of the avian influenza viruses to infect humans. First of all, I'd like to explain about the influenza A viruses. This virus has been isolated from many animal species and humans. But the source of all these viruses is aquatic birds. As previous common concept, we had believed that direct transmission of avian influenza virus to human is hard to occur because we know the mechanism of virus attachment to animal cells. That is, avian influenza virus prefers to recognize avian type virus receptor, while human influenza virus prefers to recognize human type virus receptor. For the recognition of virus receptor, the viral surface molecule HA was used. So, if the avian virus becomes infective human, the avian-derived virus must change its receptor specificity to a human virus type receptor through intermediate host like pig. Pig has both types of receptor. However, direct transmission of H5N1 highly pathogenic avian influenza virus to human is occurred. For now, 200 people were dead from this infection. At first, we suspected that the avian influenza virus changed receptor specificity to human virus type. However, the property remains as avian type. So we are confused by this fact. Why the avian virus caused disease to human? Some researchers suspected the existence of vermin. So a big question arose. Why H5N1 avian influenza virus can cause disease to human without changing receptor specificity? As second previous common concept, we have believed that influenza virus infection in humans causes upper respiratory disease. However, avian influenza virus patient showed severe lower respiratory signs. This is extremely severe pneumonia. This slide shows lung airspace filled with water, completely occupied by water. This is like drown in the air. So the second question arose, why H5N1 avian influenza virus caused severe lower respiratory disease to humans? To find the answer to those big questions, we started to investigate human respiratory tract. First, we checked upper respiratory part of human respiratory tissue. Red color shows avian virus receptor, and green color shows human virus type receptor. As you know, you can see that just only a part of nasal mucosa has red receptor, but most of all mucosa covered by green color. That means human receptor. Next, we checked the terminal part of bronchiole. Then the cells at the junction area of bronchiole and alveolar part here, you can see red color. This site cell express the avian type virus receptor. And alveolar type 2 cell also expressed avian type receptor. You can see red dot scattered in alveolar cell. So mostly upper respiratory area were covered by human virus receptor like this, while lower respiratory area has avian virus receptor. Therefore, if human respiratory tissue are exposed by avian virus, efficient attachment of the virus will occur at lower area. Attachment. But here, third question arose. Can avian influenza virus actually be infected to human lung tissue? So we tried. When we expose the human or avian viruses to human respiratory tissue, avian virus actually can infect, indicated by brown dot, to human lung cells, but just only lower part cell. Therefore, the answer to question is human lung has an environment which allows the avian influenza viral infection. In fact, the report of human victims describes the viral infection to the lung cells. So let's see the viral influenza infection to human respiratory tissue. Avian viruses are spread by poultry. The human aspirate viruses into respiratory tract. In upper respiratory area, avian virus can't reach and attach to the cell surface because of lack of receptor, weak adhesion. But in lower lungs, type 2 lung cells here express avian virus receptor, so efficient attachment, viral attachment to the cell will occur. This is receptor-intervened viral infection. And newly born viruses will appear. While human viruses are spread by coughing and sneezing, the next human aspirates viruses into respiratory tract. In upper respiratory area, many cells express human virus receptor, so efficient virus attachment will occur. Also, receptor-intervened viral infection is established. The newly born viruses appear on the surface. They must fight against ciliary motility and vigorous mucus production. But in lower lungs, the human viruses can't make infection focus efficiently. The virus grown at upper part efficiently spread by coughing and sneezing. Therefore, for efficient spread toward next susceptible person, recognition of human virus receptor at upper respiratory tissue is important. So, for now, avian influenza viruses are not efficient to spread, then can we be relieved? The answer is no. Because we showed an H5N1 virus isolated from human victim has mutation in the surface molecule, and this mutation affects on attachment to the human cells. Here. Because of the mutation, some of human-derived H5N1 avian viruses are adhesive to human upper respiratory tract. So you can see green color along here. This means green color shows viral attachment to human respiratory tissue. And without this mutation, you can see the efficient attachment, right? This is avian type, and then this is mutation. Already mutation have virus. But how can they, mutants, appear in this human respiratory tissue? We have a hypothesis to explain it. When we examined influenza pneumonia in monkey, lung alveolar damage has prominent in early stage. However, late stage, viral growth shifted to upper area. The upper area has human virus receptor. So, after severe alveolar damage, viral replication may shift toward bronchial area. How can the mutants appear? Let's observe schematic explanation. Lung is exposed by avian viruses. Bronchiole express human type receptor. Alveolus express avian type receptor. Avian receptor expressed type 2 lung cells were efficiently infected and damaged. Okay. Because of damage, lung space is filled with water. But avian influenza viruses continue to infect the receptor-matched cells. But during infection cycle, accidentally mutant is born. This mutant recognizes human virus receptor. The mutant repeats infection cycle. The mutant increases the population at bronchial area. This is our hypothesis. So, those bronchial area could be a site of viral receptor specificity change. Therefore, to prevent new pandemic, now we know that stopping of avian to human transmission and also stopping prolonged infection in patients are important. Thank you very much for your attention.